All right, so for this one, we are looking at the phases of meiosis, and so we are going to kind of talk through them. I've got some beautiful drawings here. I know this is some serious artistic license I've taken here. But we're going to go through the phases and talk about what happens in each one. So before any of this, the cell is going to be in interphase. And that's when it's just going to be preparing. It's going to be making all the enzymes that it needs to divide, all the structures like centrioles, anything, lots of extra organelles so that as it divides, it'll have plenty for the daughter cells, and so on and so forth. In prophase, what we're going to see the chromosomes condense. So it's going to go from being like an unwound skein of yarn to a nice tight skein, or what you would call the ball of yarn. And so that's when we're going to start seeing in this kind of X shape. They're also going to pair up. That is called synapsis. So synapsis is going to occur. And that's just where the tetrads pair up. So you see that the dad chromosome pairs up with the mom. Oh, another thing that happens up here is DNA is copied. So what you see is you go from having a single chromosome to having a duplicated chromosome. Okay, so you'll have 46 of these, and you go to having 46 of these at the end of interphase. All right, so anyway, then they pair up. So dad's number 1 pairs up with mom's number 1. Dad's number 2 pairs up with mom's number 2. That's called synapsis. Also, what may happen here is crossing over. And crossing over is where a piece of dad's chromosome breaks off, because mom and dad's chromosomes, as they're pairing up and all that, they're kind of getting tangled up. Okay, it's kind of a hot mess in there. And so what happens sometimes is a piece of dad's will break off, and when it reattaches to mom's and vice versa. And so what you end up with is this chromosome, this half of the chromosome on this side, now has a little piece of mom. And this half of the chromosome right here for mom's now has a piece of dad's. And so that's due to crossing over. That increases genetic variation. Cells are going to spend about 90% of their time in meiosis in prophase 1. So a lot of time there. So they just want to make sure that they're doing everything they need to do and have time to do. In metaphase 1, what you're going to see now is you're going to see these chromosomes, these tetrads we call them, because they're made of 2 duplicated chromosomes, which is 4 chromatids, and tetra means 4. So these tetrads are going to line up on this midline or equator, okay? And after that happens, you're going to start to see the spindle fibers, the kinetochore microtubules, grow out and attach to the chromosomes at the kinetochores. Remember, kinetochores are those little bracket like things on the sides of the centromere. You're also going to see some non-kinetochore microtubules come out and kind of bind to each other. And they're called non-kinetochore because they're not binding to the kinetochores on the chromosomes. All right, so in anaphase, so metaphase, they're lining up, attaching to the spindle fibers. Anaphase, we're going to start walking these guys down. So the way that chromosomes get from the center to the centriole, to the pole, the end of the cell, is that they are walked down down this microtubule with a motor protein. So this little motor protein that uses ATP is literally going to walk this guy down the microtubule. And as it does, the microtubule is going to break down, so the chromosome can't go backwards. It can only go towards the poles. So they're going to start moving in opposite directions. You've also got your non-kinetochore microtubules. They've been pushing up against each other. So they were kind of, let's see if I can do this on here. They were kind of like this, and what they've done is they've walked away from each other, and as they do, that pushes the poles of the cell apart. That's how the cell becomes elongated. So anaphase, you're seeing the chromosomes move to the opposite poles of the cell, and the cell elongates. And then telophase and cytokinesis I kind of put together. Telophase is technically the fact that these chromosomes are now at opposite ends. Some species will actually take the time to rebuild a nucleus now, others don't. Just depends. But that's what telophase. Tele means distance. So that's why you talk on the telephone, because you're talking across a distance. And so telophase just means that the chromosomes are now a distance apart from each other. Cytokinesis is where we start seeing this pinching in, which if it's an animal cell, it will pinch in, and that's called a cleavage furrow. If it's a plant cell, it's going to form a cell plate. which is just a bunch of vesicles that have cell wall material, and they fuse together and make a new cell wall across the middle, and that's cytokinesis. So telophase is the fact that the nuclei, or what's in the nuclei, the genetic material, is separated. Cytokinesis is division of the cytoplasm and organelles. Okay, so PMAT, prophase, metaphase, anaphase, telophase, is division of the nucleus. Cytokinesis is division of the rest of the cell. At the end of this point, once this pinches all the way in and forms two cells, we are going to be haploid. So at the end of meiosis 1, the cells are haploid. And the reason they're haploid is because to be haploid, you either have mom or dad's chromosome. So if this is chromosome number 1, we have dad here in this cell and mom in this cell. Chromosome number 2, mom in this cell, dad in this cell. But if you're just looking at this cell, we don't have a dad and a mom number 1, or a dad and a mom number 2. We just have one of each. And so at this point, even though the chromosomes are still duplicated, because you only have one parent, that is why it's haploid. All right, so at this point, the cell does not re-enter interphase. It does not want to copy the DNA. That would put it right back to where it started, and that would undo everything it's done. It's simply going to go straight into prophase 2. And so in prophase 2, we're going to see centrioles are moving to opposite poles of the cell. They've been duplicated again. You might start to see some spindle fibers growing out of them. Okay, getting ready to divide. Okay, not a lot of time is spent here. Not a lot's going on here. Chromosomes are already condensed. They don't have to do that. Metaphase 2, now we're going to line them up single file. And we're going to grow out those kinetochore microtubules. And they're going to attach to either side of the chromosome, both sides of the chromosome. Okay, so they're going to grow out from the centrioles towards the chromosome, and they're going to attach to those brackets on the sides of the chromosome. We're also going to have our non-kinetochore microtubules again, because they're going to be responsible for elongating the cell. Okay, so that's them. And then in anaphase, again, those motor proteins are going to walk these chromosomes, but what we've seen now is the sister chromatids have split. Those cohesin proteins are going to come through and cut all of the connections between the sister chromatids, allowing them to split, and so now they go from being 2 chromatids to 2 single chromosomes. So as soon as they split, they're no longer chromatids, they become single chromosomes. So the microtubules are going to, the motor proteins are going to walk those single chromosomes down the microtubule towards the poles. Again, they're going to go in one direction because the microtubule is going to break down behind it, so it can't go backwards. Our non-kinetochore microtubules have been pushing against each other and elongating the cell. So that's why you can see they're barely overlapping anymore. And let's do this one. And I am drawing all this in duplicate. You need to do the same on your meiosis diagram. Oops. Because your cells they've split in half, so you've got to show that it's happening in both daughter cells. Okay, and then telophase, again, is talking about the nucleus. So I did these together. You can do them together or separate. Telophase 2 just tells us that our individual chromosomes are now at each end of the cell. And then cytokinesis is that division of the cytoplasm and organelles. And this is an animal cell because it is pinching in. Plant cells can't because of that thick cell wall. So when we get done with this, what we would see is we would actually have 23 of these chromosomes. I'm only drawing 2 in the interest of time. And so you would have 4 cells, each with 23 chromosomes. So they would still be haploid. And so that is the unique processes that take place in each phase. I'm just checking to make sure there's nothing else I need to talk about. I hope that helps. If you still have questions, let me know.